Get started with our next presenting company. There's a slight zigzag to the schedule here. We're going to move up Tigenix to speak next. Presenting for the company is CEO and Managing Director Eduardo Bravo.
Thank you. And I think that someone left a very nice phone here. So great. Okay, so there's a phone here that someone left. Okay, don't worry. Nice. I will get it back to Spain with me. I would like to thank the organizers and the sponsors, and I really want to thank the rest of my colleagues that agreed that I can move up. I'm moving, and unfortunately need to fly back to Spain, and the last plane leaves JFK at 7:30, and I was going to miss it. So thank everyone for the flexibility, and I promised that I will do it in slightly less time than the 15 minutes. So I don't know how familiar you are with Tigenix. We are a public company traded in the New York Stock Exchange in Brussels. We are, I think, the only European representative here in this panel. And we have a pretty nice group of investors. Our biggest investors are actually Roche and Novartis, the venture funds of Roche and Novartis, plus three big venture funds, specialized venture funds in Europe. We have a nice free float, and we have of course, being the CEO, I can only say a very low valuation. We're worth about 120 million today, and we're covered by 5 analysts today. Why do I believe that we are not proper value? Because I think that we have one of the most advanced, if not the most advanced, pipelines in this space. We have the only cell therapy product that has been approved in Europe, ChondroCelect. This is an autologous chondrocyte that we use for the symptomatic repair of cartilage lesions of the knee. It's a relatively small market, about 20,000 patients. We obtained approval in 2009. The last 4 years, not a single company has been able to get a second product through regulatory approval in Europe. Not only did we get the approval, but also now we're getting reimbursement in different European countries, and we're currently selling in Western Europe, and we're signing distribution agreements, so far Finland and Middle East, and we're moving into Eastern Europe next. But the future value of the company will come out of the adipose-derived stem cell platform. This is an allogeneic stem cell platform of cells that come out of fat. And contrary to the cytory model that was explained, we're much more like the athersys, the pluristems, or the mesoblasts. We take fat out of a donor, and we're capable of selecting the stem cells and expanding them. From one liposuction, we get 360 billion cells, which makes it, as you can imagine, a much better business model and an off-the-shelf product. And what we've done is with this product, we've chosen 3 different routes of administration, and we are developing, as a proof of concept of that route of administration, the compound in different indications. So we have one compound, CX601, that is currently in the middle of a phase 3 in Europe for a non-very glamorous disease called complex perianal fistulas in Crohn patients. And I tell you, I also have some pictures that I don't think that you want to see, so I'll spare you the pictures. What makes this product interesting is that, being an orphan and given the experience we had in the past, a single pivotal phase 3 is all that is required in Europe to file for marketing approval, and the data is expected in 2014. So we could be filing this product for marketing approval in Europe January 2015. We're currently in the middle of recruiting in 8 European countries, 55 centers. That's the most advanced one, and we have worldwide rights of that product, so we are currently in discussions with several companies to license 601, both in the US and Southeast Asia. The idea is to keep the European rights and to find partners for developing and commercializing 601 in the rest of the world. And then the other 2 compounds are systemic delivery of the cells. 611 is the intravenous delivery of the cells for the treatment of rheumatoid arthritis. And I will not spend much more time now because that's the only product I want to concentrate myself on. Final one is 621. This is the intralymphatic delivery. So we inject the cells directly in the inguinal lymph nodes Why do we do that? Because we've demonstrated that that's where the cells need to go. So we filed a patent for that route of administration, and we've demonstrated in animal models that a fraction of the intravenous dose is equivalent in efficacy. And we believe that better in safety. So the idea is that we will keep developing both 611 and 621 for different autoimmune indications. Why adipose-derived stem cells? The great thing about going after Gil from Athers is that he always does the technical explanations. I'm not a scientist. So what I can say is that fat is a very good source, actually a better source of stem cells. It is easier to access. There are more in the same volume than in bone marrow, and they are easier and quicker to expand. Not only that, but they share with the rest of the mesenchymal stem cells the same immuno-modulatory properties that they explain. And we have an extra advantage, which are even less immunogenic than bone marrow-derived stem cells. Something very important is that we have this platform of adipose-derived stem cells, which is completely ready to go into the market. So we have first a GMP certified facility. So Sami said that they are the only ones doing their own manufacturing for allogeneic stem cells. No, we have our own manufacturing facility in the Netherlands for ChondroCelect, and then we have our manufacturing facility approved in Spain for adipose-derived stem cells. And we have those cells completely characterized. So we've been twice in front of the EMA, and we have a product today that is ready to go into the market. We have everything that needs to go on a CMC. All the release criteria, all the assays have been pre-approved by the EMA in writing. Not only that, but we have a full preclinical program. We've invested heavily in making sure that we could go into almost any indication, any route of administration in the future without spending a lot of money and a lot of time in doing preclinical work. So we did a huge preclinical program, 5 animal models, 5 routes of administration, and of course we've demonstrated that the cells are very safe. There's no toxicology, no tumorigenicity, and that they migrate to the site of inflammation. And something very important that has been mentioned by some of my predecessors here is that they do not stay very long. They do not persist. After a week, we cannot identify any further cells in the body. So my last slide, why we're going into this intravenous administration. So RA, I don't think that I need to convince anyone that this is a huge market. It's over 20 billion, and it's a huge unmet medical need. 2 examples of the unmet medical need I always give is, one is Cimzia, the UCB product, 5th anti-TNF to come into the market in this indication. Despite that, and less indications than the predecessors, is going to be doing 500 million this year in sales. Not bad. But the biggest product in the world today in the pharma market is Humira, which is main indication is RA. And I don't know if people realize that Humira is an incredibly potent and efficacious product with a black box in which 5% of the patients taking Humira will end up with tuberculosis. So that gives an idea of how serious the disease is and how much of a need there is to develop safe and efficacious treatments. So I'm not going to go through everything that we've done in animal models, in human cells in vitro. What I can share with you is the design of our phase 2A clinical trial in RA. This is the first and the most advanced and the only, I think, randomized clinical trial that has been done with stem cells in this indication. It's a pretty large trial, 53 patients that we're treating with 3 injections 1 week apart, and then we're following them for 6 months. The idea is to look at the efficacy of the cells and the persistence of the efficacy. We've been forced, because this was the first time that we were administering the cells, so primary endpoint is safety, to look at a very tough patient population. So we're injecting the cells in patients that have failed already, not only methotrexate, but already 2 of the new biologicals. And actually I can tell you, because we have the demographics, is that the average in the trial is 3 biologicals, and that we have 1 patient that had failed 8 biologicals before coming into the trial. So I don't think that there's a much tougher patient population that we can try ourselves with. And we did, December last year, we announced the preliminary safety data, which was very positive. Only 2 patients experienced some serious adverse events, only 1 discontinuation of the trial, none of the infections that you will see with the rest of the biologicals. So we think that we have a very safe. Now, is it efficacious? And that is about to be known in about a week. So unfortunately, we didn't have the data, and we don't have the data prior to this meeting, but I think that in the next week to 10 days, we will be announcing the phase 2 data in RA, and we believe that this will be a transformational event for the company. So with that, I would like to thank you, and I think that we have 43 seconds for questions.
Great. Come on over, Eduardo. So with respect to the cartilage product, tell us a little bit about the EMEA approval process and how that went.
So there were some cartilage products already in the market. We were the only company that went through doing rigorous clinical trials. So we did a randomized clinical trial comparing with the standard of care, which is microfracture. And we got a 1 year with also histology of the cartilage, and then 1, 3, 5 years data comparing double-blind the effect of ChondroCelect versus microfracture, showing superiority not only in the structural repair but also on the clinical outcome of the patients.
Excellent. Fantastic. Well, good luck with the data coming up in RA, and safe travels back home. Thanks so much.